And now I would like to introduce today's speaker. We have Dr. Kent Daane. He's a cooperative extension specialist in the Department of Environmental Science, Policy and Management with UC Berkeley and at the Kearney Agricultural Research and Extension Center. Today he's speaking on vine mealybug controls in California vineyards. So now I'm going to go ahead and pass this over to Kent. So you can go ahead and share your slides.
Thank you. So Cheryl, Peter, thanks for the invitation. To all the participants, we're going to talk today about vine mealybug controls. And really, it depends on where you're farming, what commodity you're farming, and it changes the different kinds of controls you want to use. So today's focus is primarily going to be for wine grapes, although all the control methods work for table grapes and raisin grapes as well. But I have had a little bit of a focus on mealybugs as vectors of leafroll for this talk. And I want to thank all of the co-authors, mostly postdocs in the laboratory, collaborators that I've worked with over the years. So this is a project that's been going on for almost 25 years. In fact, it's really a little bit disappointing that I'll be ending my career without really feeling like I've got great controls for this pest. But in preparing for this, I looked at some past monitoring data. And when we first started using pheromones, we were getting counts easily in the 500 to 1,000 mealybug males per week. So we have lowered the population. As an outline, we're going to start with mealybugs as vectors of grape leaf roll associated viruses, then talk a little bit about mealybug species and a part of the biology that is important for vine mealybug control and monitoring. We'll then look at three of the major control tools we've got, biological and chemical mealybug controls. And then a little bit more time on mating disruption for vine mealybug, because I think with the chemical controls, we get a lot of information from PCAs and industry. With mating disruption, there's different ways to use it. And I still find growers trying to make this work for their particular situation and going quite often under label, which can be used at different times. So we'll talk a little bit about mating disruption. And then we'll come back and conclude with area-wide programs, something that I'm really trying to emphasize these days. Don't be a vineyard as an island, because your mealybug pest problems include what's happening in your neighbor's vineyard as well. And then we'll finish off with roguing. So we start with leafroll, we'll end with leafroll. And I hope to have enough time for not so much discussion, but questions. This is a 72 slide presentation, and I picked those 72 from about 140 slides of different data and conversations and discussions. So again, the control tools don't really have to be only about wine grapes, but the wine grape growers have been more interested these days in really reducing mealybug densities down to low levels. We can see grape leaf roll associated virus here. We know it delays bud break and flowering, berry maturation, changes color of the wine, reduced sugar content, and increased acidity in juice. Basically, your very expensive wine is half or less in terms of value or price when you've got leaf roll. And I know that this varies depending on where you are. I started getting a lot of concern from growers, mostly in cool climate regions, Central Coast, Monterey, Napa, Sonoma, and even the wine grape growers in San Joaquin Valley that showed initially less response now are more concerned about it. And all growers now are worrying about grape leaf roll associated virus in combination with the vitiviruses that are causing sudden vine collapse. We did some initial survey work with members of Rodrigo Almeida's lab. Monica Cooper was involved with this, as well as Lynn Wunderlich on the coast, in the foothills. And we were looking at the different species or groups of leafroll viruses that we were finding in the state of California. And as in many regions around the world, Leafroll 3, green in the pie charts, dominated what we were finding, curiously, except for the vineyards in the Sierra foothills, where we had Leafroll 2. Now, Leafroll 2 is that one group where it's not really known to be vectored by an insect or a mealybug. It would be great to get the same kind of work done in Southern California and in the San Joaquin Valley proper, Merced, Madera, Fresno, Kings, Tulare, and Kern as well. And you can see a gap there too in Monterey and Sonoma. So it'd be great to fill this map in and know exactly what leafroll group we're dealing with. For most of these different strains, an insect is the vector. Again, Leafroll 2 doesn't fall into this group. For most of us, we're concerned about mealybugs, but the Pulvinaria scale, the European grape scale, or the European fruit licanium scale, these are also vectors. And talking with colleagues in France, they feel like these are at times more important vectors than the mealybug, which we'll get to later. I suspect it's because they don't have our vine mealybug there. I show pictures here of the vine, grape, obscure, and long-tailed mealybug. I do that because it doesn't matter which mealybug you've got. They tend to all be vectors of leafroll. So this is just a association chart. Don't worry about the mealybug species names on the right. And notice that for the leafroll species or strains, most everything of these connect the dots are going to Leafroll 3. That's just because Leafroll 3 is the most common leafroll group around the world. And so most of the studies have been done with that as well. What I want to point out is these arrows are going all over the place. There's no apparent fidelity. All of these different mealybugs can vector any of the leafroll strains, except for 2, that we appear to give them. The other thing I want to point out, which I'll talk about a number of times, is that in California, we pretty much have all of these mealybugs that have been found to be important as vectors of leafroll around the world. Now, there are some differences in the efficiency of these different mealybug species to be vectors of this. So examples here, it shows that the long-tailed mealybug, Pseudococcus longispinus, was perhaps the most efficient vector, followed by vine mealybug, Planococcus ficus. A lot of this, though, drops out when you think about the mealybug biology in the field, because the vine mealybug has so many more generations per year. So yeah, there's a difference in a lab when you're moving mealybugs with a brush onto a vine, but that drops away when you look at biological differences in the field. One of the most important aspects of vector biology in terms of being transmission is being a vector. The crawlers, and this work was done with the grape mealybug, but it's the same for most of the mealybug species. The crawlers could acquire the virus, feeding on an infected vine, pick up the pathogen within 1 hour, and then transmit the virus within 1 hour. In our study, there was a peak at 24 hours. Crawlers were the most efficient, but all of the mealybug stages could acquire and transmit the pathogen for leafroll. Another important aspect of their vector efficiency is that it was a semi-persistent transmission. So basically, they lost the ability to transmit the pathogen every time they molted, and after about 4 days. This is probably because it was being held inside their esophageal gut system or their mouth parts. It didn't make it into the insect's hemolymph, and it wasn't persistent within the body. Something we're always asked by growers is how quickly can I see it in the field? So what we did here is we took the grape mealybug and we put it on, over in the middle picture on the right, we put it on infected vines in the glasshouse. After a 48-hour acquisition, we took those insects, mostly first instars, we just cut off the leaves, and we took it to the field and we put them onto a single vine. So 10 mealybug first instars, 48-hour acquisition, 48-hour inoculation. We then treated that vine with pesticides frequently, so we knew it was our inoculation. We did this around the 1st of July, and we saw no symptoms that year. Let's look now at the second year. Boom, in June, we start to see symptoms. Third and fourth year, we're seeing symptoms mid-April and into June. And usually about 30% of the vines were symptomatic, meaning that we're about 30% efficiency with just 10 first instar grape mealybugs on that vine. So this points out, first, you don't see the infection that first year. And we were inoculating in July. If the inoculation event is in August, September, certainly you're not going to see symptoms, but that vine has now the pathogen. It's a diseased vine. Second year, a little bit less of a little delay, but by the third year, we're seeing symptomatic vines by about June. Let's switch over now a little bit to California mealybug species, even though today's talk is really going to focus on vine mealybug controls. There's the vine mealybug. It's one of about 8 invasive mealybug species in California vineyards. It's from the Mediterranean region. We know that, and I'll show you some details later. It made it to South Africa about 60, 70 years ago. California and Mexico about 25 years ago. Then we knew it was in Argentina. Then I knew it was in Peru. Then Brazil. Then Oregon. And I suspect now it is also in Chile. There's no reason why it's not going to make it to Washington. There's no reason why it's not going to make it to Canada or the East Coast of California, or sorry, the East Coast of the United States. It causes more damage than the other mealybug species because it has more eggs per female. It has more generations per year with more eggs per female each generation. Feeds readily on the leaves, but we'll talk in detail more about that. And since it's developing so much faster compared to the other mealybugs, more honeydew, more excretion, more direct damage. This is important for raisin growers, table grape growers, as well as wine grape growers. Especially more damage compared to our native grape mealybug. It's one of the first of a number of Pseudococcus species. There's also the obscure mealybug, Pseudococcus viburni, from South America. Long-tailed mealybug, Pseudococcus longispinus, from Australia. Note that you can't use these filaments, these tails, always as an identifying feature, because when they molt, they lose that white waxy tail and filament. And so you have to wait for them to get that white waxy back. Another Pseudococcus is the Calceolari, the Citrophilus mealybug, also in California, from Australia, one of the most important mealybugs in New Zealand. Here in California, not so much a pest. Pharesia mealybug, a different genus, a different group, probably from southeastern US, Jamaica. Citrus mealybug, or other Planococcus species. An important vector of Leafroll in Spain, not so much in California. Maybe their citrus mealybug is a slightly different strain. And that probably is what's going on with our vine mealybug. This is a family tree of the vine mealybug. Here's ours. We know ours is from Israel. Each of these little hash marks is a greater distance. So we can see the orange dot right here next to California and Mexico. California and Mexico are so similar. It was one invasion event. Israeli population. Next most related is Egypt. All of these little bars here suggest that these populations in Europe are very far apart from us. And you can see that the South African population probably, and the Argentinian population probably came from trading material with Europe. And we've got this other population, Southern Italy, Turkey, Tunisia. This will become important later on in this presentation. What's important right now is we've followed that initial work up with a deeper dive into the molecular aspects. And we know that our North American group probably is a different species than this European South African group. So Planococcus ficus from the fig tree is probably what they've got in Europe and South Africa. Planococcus vitis is what we've got in Israel, North America, and it's related to what's in Egypt to some extent as well. That becomes important when we're looking up controls and looking at future controls. So for example, this is the relationship of our different groupings to pheromones. The pheromone we've got is Lanaval senesioate, and that's in blue. We're all responding to that. California, Iberian Peninsula, southern Italy. But look here in the Eastern Mediterranean, there's a group that responds to that, a group of males, but a group that doesn't respond, that's indifferent. And we get that in California as well. And a group that responds to another kind of pheromone. So even though everything's been grouped as Planococcus ficus, we've got these other populations. And we've got populations in California, Europe, and Sicily that don't respond as well to our pheromone all the time. The most important difference between our vine mealybug and, for example, the native grape mealybug, to me, is the number of generations per year and the fecundity. So while we're getting in the San Joaquin Valley between 5 and 8 generations per year, and with milder winters, even more, grape mealybug up here in the upper left only has 1.5 to 2 generations per year. So whenever we see studies from New York that we're going to see later on Rogan, remember they're dealing with a different pest. Quick description of its seasonal biology. We've got the vine. Winter and early spring, most mealybugs are on the trunk, underneath the bark, sometimes on the cane, depending on where you're located, also on the roots. It varies by location. So in Bakersfield and Coachella, bigger population on the roots, smaller population, but some still in Fresno. When I go to to Central Coast, San Luis Obispo, Napa, mostly on the trunk underneath the bark, less underneath the bark on the roots. As the season progresses, they follow the phloem, they follow the sugars, they get onto the canes. Early summer, we used to see these on the leaves, not as much anymore, and then into the fruit clusters. I just mentioned that we used to see more of the mealybugs moving out to the leaves by early summer. We're seeing more of these staying underneath the bark and then going into the fruit clusters in July, August. I think a lot of that has to do with all of the neonicotinoids we're putting on, the Movento applications that really go out to the leaves. So I think we've kind of forced that mealybug now to be more of a trunk cane mealybug until those neonics wear out of the system, we're off the vine. The Movento wears out of the system, off the vine as well. Monitoring, you can look for damage, you can look for drop leaves, you can look for ants, you can strip bark and look for mealybugs. That's all very labor intensive, it's all very tedious. I much prefer using pheromone traps. I've heard from growers that say, I've got a mealybug problem, I know I'm going to spray, so I don't waste my time with pheromone traps. I say baloney. Use those traps even if you're doing it towards the end of the season. August, September, October, when we start to get heavier flight numbers. Put it out there and just check it once or twice in September to get a population density record that you can go then back into history and say, well, this year I put on a delayed dormant applaud. I put on a neonic. And next year I had higher, lower changes in the population based on the pheromone trap captures. So I like to use it to keep that historical record. There is a second movement looking at sniffer dogs, and there are some dogs being trained right now. I talked to Neil earlier this week that's working with this group and said in the laboratory, the dogs were on scent, both for vine mealybug and leafroll using potted plants, and with some limited outdoor trials. They were taken into Russell Ranch, where they know there's leafroll. The leafroll virus dogs tested were a little bit confused in the trellised vineyard system, but they're going to work on that this year. And they're about 1 to 4 months out to start to get some field effectiveness tested. So now we're going to move into controls, which is effective for everybody. I am not going to spend a great deal of time on biocontrols. We've got our predators, green lacewings, mealybug destroyer, cecidomyid midge. This is one of our native mealybugs, native beetles. That I think is a skimnus, lots of different native beetles. They're all great. They all tend to have one downside, and that's that they tend to be in the vineyards more when the vineyards have a lot of mealybug. They don't tend to work great at low mealybug densities. The natural enemies that tend to work better at low mealybug densities are the parasitoids. And the big picture is an Anagyrus, Pseudococcus, or Vladimiri, adult female, spends most of her life inside the mealybug with her mouthparts, chews a hole to get out. We can see the adult male in the lower right. And in the upper right, we see a parasite that we got from South Africa called Coccidoxinoides peregrinus. Okay, so the Anagyrus is by far the most common, by far the most important. It has got one critical downside. Even though in organic vineyards, I've seen this parasitizing as much as 80–90% of the exposed mealybug, its downside is that it always starts late. And so we're starting to see parasitism typically in June. And that's because of this. We see here mealybugs, and when the parasite emerged after the oviposition date. So we exposed mealybugs on potatoes, and the black bars all the way along here is when we took half the group inside to 78 degrees. So you can see the black bars went from egg to adult, always in about 3 weeks. The group that we left outside, exposing in October, November, January, December, February, March, and April. Regardless of when we exposed them, when we left them outside, they didn't take 3 weeks to emerge. They took up to 6 months. They always came out in May. And that's because the mealybug doesn't care what you need, it cares what it needs. The parasite, it's going to have this winter hibernation, winter estivation, because if it were to emerge in December or January or February or March, even April, most of the mealybugs are still underneath the bark in a protected location. And so the parasite emerges as the mealybug is starting to emerge from underneath the bark and starting to go out and follow the phloem, where the mealybugs are starting to go out following the phloem to the leaves. So there are some ways that we're looking at to get around that. We've done releases by hand in the past, and we found large releases of Anagyrus, smaller releases of the mealybug destroyer were able to lower mealybug densities. But here what we're doing this year is we're testing drone releases. In the first year, last year, we found that it wasn't very effective with the mealybug destroyer because we were releasing adults and they were just flying away. So this year we're repeating releases with the Anagyrus vladimiri wasp to see if we can find any reduction. I'll note that when we did it by hand, we were releasing as many as 5,000 per acre as a proof of concept. And it did work, but that's economically too high for most growers to afford. So what about our classic biocontrol program? We initially imported Anagyrus psittacoxy from Egypt, Israel, and we even worked with a colleague in Iran that reached out to us. But that's in orange because that material, there were so few, we never got it out of quarantine. We got the Coccidoxinoides peregrinus from South Africa. Then we got Leptomastix dactylopi and Leptomastidia amnormis out of Europe and released that. And we got Anagyrus, at the time, psittacoxy, what we now know to be vladimiri, from Spain and Sicily, and released that. So I just mentioned this difference between vladimiri and psittacoxy. And you're probably wondering, because if you're purchasing this, some people are selling psittacoxy, some people are selling vladimiri. It's really hard to know what you're getting. These are very, very similar species. It wasn't until very recently that our colleague Sergei Trapitsin separated these out, calling one psittacoxy, calling the other vladimiri, based on some antennal differences and some molecular differences. As far as I can tell, the main difference is that what we were importing and releasing, vladimiri, it will attack smaller mealybug stages, whereas what was in California before, psittacoxy, and is still here, attacked typically the larger mealybug stages. So let's look at what we've got, and this opens up for more work in the future. We've got this Leptomastix dactylopi and the Anagyrus psittacoxy, or maybe vladimiri, that likes to attack the larger mealybug stages, Leptomastidia abnormis and Coccidoxinoides peregrinus, that likes to attack the smaller mealybug stages. And we're throwing this Anagyrus vladimiri into the mix that I got from southern Spain, or southern Italy and Spain, which also tended to attack the small stages as well. Remember, I talked about the differences in mealybug. What's exciting to me about that is that if you look now at the mealybugs in the Mesopotamia area, which may be where our vine mealybug originated, we've got parasites that we have not imported attacking their mealybug on vines, probably this Planococcus vitis. And so we've got 4 species there, Anagyrus, all Anagyrus that we've never imported. And that's one of our goals in the future, is to bring these in. And this is shown in the work I did with Majid from Iran. He does get this Anagyrus pseudococci as his most common species, but their Leptomastix dactylopi was also extremely common. And these Anagyrus agrariensis, dactylopi, and nizer, I'm messing up these names because I don't work with these, were also fairly common. So that's a goal for the future, is to bring these in. Now, one of the aspects of natural enemies is they are easily disrupted. They're disrupted by insecticides. They're disrupted by ants. We've seen this movie in the past where we've got an ant lapping up the honeydew in front of a mealybug. A parasite just put an egg into that mealybug that was Leptomastix dactylopi, or sorry, Leptomastidia abnormis is the one that is the parasite back there. It is in California. I occasionally recover it. Down below, that's the mealybug destroyer. And we know that ants protect the mealybug because they defend against the parasite. And we see that in this often played video here, where the parasite comes back and the ant, now on guard, kills it. So to have some of these parasites work well, we have to control the ants. Monica Cooper and David Haviland and Mark Cottle have led different research programs looking at hydrogels. Postdoc in my lab, Nathan Mercer, working with David's group, has tested this in the Lodi Woodbridge region. David's tested this in the Bakersfield table grape region. And we've tested in the first year different active ingredients, agromet, boric acid, esteem, and trust platinum, mixed with 25% sugar, water, very low label rates. Very low rates, so 1/100th to 1/200th of the label rates for these products. And we've had very good results. All products were lower in cumulative ants at 5 gallons per acre and 10 gallons per acre. But platinum, thiamethoxam, in particular, was very good. And when we applied 2 ant control doses, platinum was great throughout the entire season. So we're continuing that work this year in table grapes, David Haviland, and wine grapes, myself. And Monica's also cooking up new cool ant controls as well. So even though we want to control ants, we want to do all kinds of things to support natural enemies. We have looked at cover cropping for mealybug natural enemies. And while it does help with the gentlest predators, like the beetles, like the green lacewings, remember that these parasitoids are specialists. There's no pest in the cover crop they're that interested in. And they get their nectar from the honeydew of the mealybug. So while I support cover crops for soil health to attract generalist predators, they may not be that great at supporting the specialized parasitoids of mealybugs. What might be even more important is selecting the pesticides you use, because even some of our organic materials are harsh on beneficials. So if you're purchasing natural enemies, think about what you're spraying on and try to coordinate those so you don't release when you've got a spray coming up that same week. So what about pesticides? This is when I started working with Walt Bentley. We were using mostly lanate and Lorsban in season. Lorsban was being hold. Lanate's now gone too. And so Walt got a Section 18 and then later got permission to use Lorsban as a delayed formant. So all of these mealybug control programs, Lorsban, applaud as in-season, and the neonics. So the neonics and applaud in-season, all that's being questioned a little bit now. And so we have to always be looking for novel materials and new ways to use these other compounds. We've tested pretty much most of the different compounds that have come out there. In most of our trials, the Movento spirotetramat has been our best product. We've got our control up here, Movento down below. We'll look at Savanto as a drip here. Savanto as a drip applied back here in May when June, when we were putting on Movento. Gave us pretty good control, started to break down a little bit after August. We remove Movento, we'll add Savanto as a foliar, which was applied right here in early July, and it gave us pretty good control going out into October. So I bring this up because we have so many different pesticide tests, and I find them to be very regional, to be very site-specific. What worked great for me on an old raisin vineyard may not work as well on a young wine grape vineyard in Napa. Mealybug movement changes with the season, changes with the heat, changes with the vine physiology, changes with the irrigation practice you're using. Irrigation practices change how the neonics work. Your PCA, the PCAs know best what is working well in your area. Soil type changes how the systemic neonics are taken up by the vine. So be your own researcher. Yes, talk to us, look at some of our work, but work with your PCA to find out what products might work best for you and might work best for your budget and your level of vine mealybug. We've spent a lot of time, especially working with Luca Brilliante at Fresno State, looking at some organic products because we wanted things that would work well with our organic options, especially in area-wide programs. What can we give the organic grower? The common issues with some of these products is they've got little residual activity, so they need repeated sprays. They target smaller stages, and so you have to go back again to get the larger stages. And they tend to be broad spectrum, so they are sometimes harsh on beneficials. We get a lot of questions about rate of spirotetramat. Should I go on 8 ounces? Should I go on 6 ounces and then later in the season 6 ounces, or 8 ounces later in the season 4 ounces? So here we did a trial where we actually went over rate. We put it on at 8, 12, and 24 ounces. And with an HPLC, we looked at the amount of spirotetramat the day of the spray, 7 days later, 30 days later. What I'll show you here is that the amount of spirotetramat increased with our increasing application dose, and it decreased over time. That's exactly what you would expect to happen. Everything looks great. Look at the amount of enol in this same vineyard. 8 ounces to 24 ounces 1 day after spray. It sort of looks right, but the 12 ounces seem to be a little bit less. At 7 days, we actually had more because probably it all had not metabolized into the leaves yet. But at 8 ounces, it was higher than at 12 or 24 ounces. And most everything was disappearing by 30 days. So it didn't quite fit the mold. I say this because I've seen different sprays of Movento react differently in different vineyards. So I don't think 8 or 8 and 4 or 6 and 6 make as much difference as working out what works well in that vineyard in terms of spray dates, irrigation levels, and vine condition. So again, work with your PCA to find out the application timing and rate that works best for you for Movento. One of the aspects about both Movento and many of the neonics applied as a systemic is that they don't get a lot of material to the trunk. Here we're looking at the amount of spirotetramat enol, that's what kills the mealybug, on 20 different vines. And this is looking at 5 days after treatment, leaf petiole samples, on average about 6-7 parts per million. And we're saying that every single sample, every single vine had some level of enol. So Movento did its job. We're looking here about 30 days later at trunk samples, and the average is about 0.5 parts per million. So about a 100-fold reduction. What's more important is that we see in 5 samples out of 25, we're getting spirotetramat enol. In 20 out of 25 samples, we're not getting anything. So on the trunk, it's a hit or miss, and that creates a window, a refuge, where the mealybug can go and feed and not get impacted. So we spent a lot of time looking at chemicals to control, and here's looking at Meyer, Movento, Sequoia, Savanto as a drip, Savanto as a foliar, vine mealybug per trunk, nothing great. When we had 2 products, Movento followed by Sequoia, so Sequoia followed by Movento, and Meyer followed by a sale, Movento followed by a sale, much better. So I think now we're in the realm where we have to consider in some vineyards 2 modes of action, 2 applications to control the mealybug on the trunk. We have tried different surfactants. So here we've got this carpenter moth with mealybugs. There's the moth, there's the mealybug. On the right is the adult moth found in Fresno area. We tried Savanto for the mealybug, Vexer, which is Intrepid, for the moth, and we tried it with Pentrabark. We saw all of the chemicals reduced the mealybug or the carpenter moth, but the Pentrabark didn't make any difference. So we're still looking for something to have an impact to get the product underneath the bark to control the mealybugs. One of the applications that did seem to have a beneficial impact was a delayed dormant application of applaud. So this was put on when we had about 6 inches of growth. So it's really not a dormant application. Lord's was dormant. The applaud was about 4 inches of growth. So now we're into early March, but no residue on the fruit at all. And we can see the control and our applaud at 12 ounces. Our applaud at 24 ounces was actually better than the Lord's application. So I'm going to conclude now, kind of going too quickly, but we'll look at mating disruption and area-wide programs and roguing. We've worked with most of the different products. We haven't worked with the Semios aerosol, but we have worked with the Sutera products, the Tracy products, and Pack Biocontrol. I like them all. Choose what's working best for your vineyard. Just pictures here. We've got the adult male mealybug next to a female putting on an ovisac, adult male mealybug copulating, female mealybug up here on top, down below the skinnier, longer, immature mealybug. We see here how mating disruption works. That is a sliver of a rubber septa for monitoring mealybugs. And we've got the male mealybug spending 2 hours trying to copulate with this rubber septa. Nathan Mercer took this video. We've got a female virgin female mealybug up here, and this adult male ignored that female mealybug until she bumped into it. and then it mated and then went back to the rubber septum. So it is the female mealybug that's putting out the pheromone. Work out of Riverside and Jocelyn Miller's lab shown that it's on. This is an upside down drawing of a mealybug. This is the hind coxa. On the immature, you don't see these pores. On a mature female mealybug, you have all these little pores on that hind coxa, and that's where the female emits the pheromone from. And so how mealybug mating disruption works is that we're overwhelming the system, the vineyard, with so much sex pheromone that the small amount this female is putting out is just not picked up by the male mealybug. So we started this program with Jocelyn just looking at developing pheromone lures, and that became commercial in the 2000s, 2002, 3. We then started doing mating disruption trials in 2003, 4, and commercialized this in 2008. And there was this period when we thought adoption was never going to take off. A number of things happened. Farm advisors David Haviland and Monica Cooper started working with this product and started really doing very good outreach. David really focusing on the sprayable, Monica on the plastic or on the passive dispensers, which is why we saw more sprayable down south, more dispensers up north, most likely. But that really did change. Along with that, we started having not just Sutera products, but Tracy and PacBio and Semios as well. And so we now are seeing an increase in mating disruption use. So how do you use this? So this is looking at pheromone trap captures, and we can see here mating disruption with Pacific Bios passive dispenser shut the traps down throughout the season. Application of Movento here knocks the population down, starts to recover a little bit as the Movento application wears off. What we're going to now do is to look at this on a plot-by-plot basis, because this is how I think one of the important aspects of mating disruption. So it was an 80-acre field. Wind was coming from the north going to the southeast. Control plots are in yellow. Mating disruption plots are in red. We can see before the application of mating disruption, we had a hot spot right here in the middle and a hot spot over on the eastern portion of this block. At the end of the season, our hot spot was now over on the west. All the mating disruption were in cool blue or green. The control here had a little bit of mealybug activity going on. So plot size, I think, does matter. While mating disruption can work on an individual plot, small plot, individual vine basis, the larger the area, the more benefit you're going to get. Also, it works better year after year after year. So this is the second year. We still have got this hot spot up here, but everything below is cool blue at the beginning of the season. So these controls are getting the benefit of being surrounded by mating disruption. Bottom graph, end of the season, just confirms that. The only little spot in the control was upwind. So plot size, wind direction, all these elements are important. Mealybug density is important as well. So just imagine a male emerging amongst all these females doesn't have to use the pheromone to find the female. So what we did in this graph is in one vineyard, we rated the density of every single vine, applied mating disruption, then at the end of the season, looked at the per capita change. When the vines were rated as having low mealybug density, we were getting about a 90% reduction. A medium mealybug density, about 45% reduction, and almost no reduction with a high mealybug density initially. And here's one of the studies we did with the sprayable. So one of the downsides of the sprayable is that you really need to put on 5 applications to have season-long control. And we see that the more you put on, the better it is. So our control is in solid circles, an early and mid-season application in open circles, starting to break down a little bit by October, and an early, mid, and late season upside down triangle started to lose a little bit in September, October, because our last spray is in August, but gave us pretty close to trap shutdown for most of the season. So with knowing how to use mating disruption, having different products, having Monica, having David, having others, talking about this application, look where we are now, this huge increase after 2016. We're over 200,000, and it's probably much higher because a lot of growers don't realize you're supposed to report mating disruption in your pesticide use reports as well. So it's increased not just in California. We went from looking at mass trapping in Israel of the citrus mealybug to a little bit of studies going on in California to now mating disruption being used throughout the world. So I'm just going to blast through these slides on roguing and area-wide control. Sorry, I took too much time. There we go. So area-wide control is important. It includes monitoring to know where the mealybugs are, mapping a region so you know not just what you've got, but what your neighbors have got. This is being done in Napa through Monica's program. One of the beneficial aspects of this is looking at area-wide program from the second year to the third year when it ended, and we see damage increased when we stopped doing the area-wide program and pesticide use went down. This is because we no longer are telling the growers, this is your population. There's no longer pressure from growers to spray to help protect their neighbors. And so I think one of the best aspects of area-wide is communication amongst people within the region. What are you spraying? When are you spraying? What is working? What is not working? For Leafroll, roguing is key. It works in South Africa. It works in New Zealand. It worked in Napa. It worked in New York. Okay, so roguing is key, but it's not applied everywhere. One of the aspects is that it's very expensive. In New York, what they did in this study is they removed the infected vine and they removed 2 vines on either side. And in New Zealand, they even removed 2 vines on either side and the adjacent vine across the way. There are other impediments because you have to survey and you can't possibly pay to test all of these vines. So you're surveying based on vine appearance, vine color, and that's hard to do with some cultivars. You can have red blotch as well and not realize that. Leafroll symptoms don't always show as red. Another impediment found throughout is that the initial disease incidence or the mealybug density impact the roguing effectiveness. So if you've got really high levels of mealybug or really high incidence of leafroll, then roguing is going to be more expensive and less effective. Also, depending on where you're located, this is work in Central California, great cooperators. They're roguing from 2019, 2020, 21, 22, they did not, and 2023, they get this increase in incidence because of that. So they're roguing, but they're not seeing this huge reduction. And the year they stop, there's a lot coming back. So one of the aspects I've found is that the examples where we see it's really working great, for example, Napa, some of those growers might have a greater resource and expense budget to rogue compared to growers in other regions of California. The other major difference I've seen is that a lot of the work we saw in New York, Napa, New Zealand, that was with other mealybug species. This Planococcus ficus or vitis that we've got now in California might just be a different animal. people are working on control tools to make this easier, such as spectrophotometry to identify diseased vines. And if you want to see more about this work, I would go to Monica Cooper's Napa Viticulture Team website. She's got a lot of great information there. So I went 3 minutes over. I just summarize quickly. None of the mealybug controls are 100% effective, not of the insecticides, not the mating disruption, not the natural enemies. Biocontrols are always present. There may not be enough of them for control as vectors. No single insecticide is 100% effective. Trial results can vary, and as David Haviland mentioned, consistency is important. I think mating disruption should be part of a vine mealybug program. I think it works best at low densities. You lower that mealybug density, and you could probably lower the dispersal rate you put out, saving money. I think area-wide control is great. I think working with neighbors is great. Leafroll control is harder because it involves roguing, and there's an economic issue in some regions. So it's not always economical, but especially when you start planting, start clean and try to keep clean. So with that, I apologize. I went 4 minutes over, and I can stay for questions if there are any. If not, email me questions and remind me you saw the talk, and that's where you're asking questions from. Yeah, I can just quickly, a lot of the pesticides I mentioned do qualify for organic status. You can look up Google Omri pesticides. At vineyards where there hasn't been any vine mealybug found, should I put out, absolutely put out pheromone traps to monitor for vine mealybug if you haven't found it, if you don't think you've got it. Absolutely do it. Bread, coccidoxinoides, is it promising? You know, I think it's more promising than we realize because it's hard to find it. It causes the parasitized mealybug to drop off the vine. So with the other parasites, you can see the mummy. With the coccidoxinoides, it's out there, but it's hard to sample for it. It's not out there as much as I think anagyrus is still the best one. The gel beads are not commercially available. They're still not DPR approved. David Haviland is working on getting them commercially registered. And once that happens, we think there's probably going to be a group that works on getting it commercialized. And then Mark Hoddle's group is working on material that might actually be organic. Our hydrogels are not. I think Monica might be looking at something that thing as well. James, thank you.
Thank you so much for answering those. Thank you again for your presentation.
Great, thank you.
Thank you, everybody, for attending today. And thank you again, Kent, for your presentation. And we hope we will see everybody back in our next webinar in September. All right, have a great rest of your afternoon, everybody.